Okay, hello everyone. My name is Leanne Black and I'm a lecturer in pharmacology on the MPharm course, and I'm going to be teaching you microbiology this term. So first of all, what do we mean by microbiology? So microbiology is the study of microscopic, so very tiny organisms such as bacteria, viruses and fungi. So there'll be 8 lectures on microbiology, and these will focus on all the essential information that you need to know as a pharmacist. So hopefully it should be really interesting for you guys. What you learn this term is critical for you. So it basically gives you a solid foundation of knowledge that you will then build upon when you study infection and immunity in the 3rd year. And I just want to say before we start, if anyone's got any questions, they can email me at the email address shown here. There will be 2 tutorial sessions to go through exam questions. So let me know if you have any queries, topics that you want to go through in the sessions. Also, if you're struggling at all with any of the topics, you can email me and we can always arrange a one-to-one session to go through things. Okay, so for today's lecture, we're going to be talking about bacteria, which is a very important topic for pharmacists, as there are many clinically significant bacterial diseases. Okay, so what will you learn in this lecture? So by the end of the lecture, you will know what bacteria are and how they're classified, as well as the different components of bacterial cells and their functions. You'll also know how bacteria move from one place to another and how they reproduce, which is a process known as binary fission. We'll also look at the staining techniques used to identify bacteria, such as gram staining, which you may have heard about. Then in the next 2 lectures, we'll look at bacterial growth and bacterial diseases. So a tip for you guys is to pay special attention to the words highlighted in blue in the lecture. So it's really important that you understand these blue words when it comes to the examination. Okay, so pay special attention to those. So to start off, we're going to talk about the main features of bacteria. So a little bit of history first. Bacteria were first discovered by a man called Leeuwenhoek in 1676 when he was using a microscope to actually examine the plaque he'd scraped from his own teeth. And he originally named the organisms he saw as animalcules, which literally means little animals. So originally they thought they were animals. So this term was then changed to bacteria by Ehrenberg in the 19th century. And bacteria means little stick. And they were called this because a lot of bacteria are rod-shaped, and so under the microscope they look like little sticks. And just a note on terminology, so we use bacteria as the plural term, whereas bacterium is singular. Bacteria are actually unicellular, prokaryotic microorganisms. So unicellular means they only have one cell. Prokaryotic means that they do not have a true nucleus that contains their genetic material. Instead, the genetic material is simply present in their cytoplasm. Okay, so this is in contrast to eukaryotic cells, such as animal and plant cells, which have a nucleus surrounded by a nuclear membrane. So bacteria size ranges from 0.5 to 5 micrometers, so quite big size range. And they come in different shapes. So there are 3 major shapes of bacteria. We mentioned just previously about rod-shaped bacteria, the little sticks. These are also called bacilli. And you can also get round or spherical bacteria, and these are called cocci. In addition, you can get spiral-shaped bacteria called spirilla. And the bacterial cell is actually covered by a cell envelope. And this is in contrast to cells such as animal cells, which are only surrounded by a cell membrane. This bacterial envelope consists of the plasma membrane, the cell wall, and the capsule, although the capsule is only present in certain bacteria. Okay, and we'll talk about all those components in more detail later on. So as we said previously, there's no nucleus in the bacterial cell, so the genetic material is just present in the cytoplasm. The region of the cell that actually contains this genetic material is called the nucleoid, but as we said before, this is not surrounded by a nuclear membrane. Some bacteria actually have extra chromosomal DNA. So this is DNA in addition to their main genome. And these are called plasmids. And these are small, circular, double-stranded DNA molecules, which we'll discuss in more detail later on. The bacteria also contain ribosomes, which are for protein synthesis. And in addition, they have mesosomes, which are foldings of plasma membrane within the cell and are important for cell division. Bacteria have also several appendages on their surface. The main one is the flagella, well, flagellum singular, flagella plural, and these are long appendages on the surface of the bacteria and are critical for bacterial movement. So it's really important for you to remember the word flagella when we're talking about bacterial movement. Another important appendage is the pilus singular or pili plural. Pili are important for transferring DNA from one cell to another. And again, we'll discuss that later. Bacteria usually occur as gram-negative or gram-positive. And using bacterial stains, we can differentiate between these two types. And we'll have a section on staining of bacteria towards the end of the lecture. Bacteria actually have an absorptive mode of nutrition. So bacteria don't have a mouth or a gut like humans. So they absorb small organic molecules, the nutrients, directly from their surroundings. And the way that bacteria multiply is by binary fission, which is actually a form of asexual reproduction. And this is where one cell splits into two cells. Some bacteria produce spores in a process known as sporulation, and this is actually crucial for the survival of certain bacteria and some actually clinically significant bacteria as well. So this slide talks about the taxonomical classification of bacteria. Okay, so there are 8 distinct taxonomic categories. These are domain, kingdom, phylum, class, order, family, genus, and species. And with each step down in classification, organisms are split into more and more specific groups. So you don't need to worry too much about the taxonomical classification because you're not doing a microbiology degree, you're doing a pharmacy degree, but you do need to be able to determine the genus and species. So this slide looks at the example of E. coli, which is a highly significant bacterium. You've probably heard of this because it's well known for causing food poisoning. And if you look at the order and family of E. coli, you see the word entero. So entero means lives in intestine. Okay, so that makes sense for E. coli, given the disease that it causes. Just to note that with the genus, the first letter is capitalized, whereas with species, it's in lowercase. And the convention is to put the family, genus, and species in italics when you're writing about them. So the size of bacteria ranges from 0.5 to 5 micrometers and can be seen under the light microscope. And this scale just puts it into context, really. So you can see that the atom is the smallest on the scale at one end, and then you've got pollen, which is obviously visible to the naked eye, human egg and frog egg at the top end of the scale. Then you've got bacteria in the middle. This just puts it in context for you. So earlier in the lecture, we looked at the 3 main shapes of bacteria. So we looked at the round cocci, the rod-shaped bacilli, and the spiral-shaped spirilla. But there are also variations on these main shapes. So there are other rod-shaped bacteria. So there's the curved rods, which are the vibrios, plural, vibrio singular, and shortened rods, which are the coccobacillus, singular, or coccobacilli, plural. And you can also get another type of spiral bacteria. So these are helical spirals, and these are called spirochetes or spirochete, singular. Okay, so we know that bacteria grow in colonies and form arrangements. So, for example, a pair of two cocci form a diplococcus, and then four cocci in a square is a tetrad. The chain of cocci is streptococcus, and you may have actually heard of strep throat. This is a common throat infection caused by group A streptococci. So as I said, streptococcus is a chain of cocci, whereas staphylococcus is a cluster of cocci. So you may have actually heard of methicillin-resistant Staphylococcus aureus, commonly known as MRSA, which is what we call a superbug, as it's resistant to many different antibiotics. So really difficult to treat, really serious problem in hospitals in the UK. And this infection could be extremely serious, even resulting in death. So you'll cover more about antibiotic resistance later on in the course. Okay, so it's a very big issue, and pharmacists have a big role to play in preventing it, because one of the major causes is overuse and inappropriate use of antibiotics lead to this resistance. Okay, so pharmacists have a big role in trying to get antibiotics prescribed properly and to get people to take them properly. Okay, so you'll study more about that later in the course. And then at the bottom of this table on the slide, you can see streptobacillus, or streptobacilli for plural, and this is the name we give to a chain of rods. Okay. So this slide shows a very good image of a bacterium and all its different cellular components. And we're going to go through all of these components now, one by one in more detail. First of all, we're going to talk about the capsule. So some bacteria have an additional cell envelope structure called the capsule, and some of the pathogenic bacteria in humans have this structure. So the main component of the capsule is the glycocalyx, which literally means sugar cup, and this is made up of polysaccharides or proteins, or a mixture of the two. And it's important for you guys to remember this word glycocalyx. Based on the presence of the capsule, we can classify bacteria into capsulated, so with a capsule, or non-capsulated, without a capsule. The capsulated bacteria will form smooth colonies because of the capsule, whereas the non-capsulated bacteria produce quite rough colonies. So when they're growing on plates, you can easily distinguish between the two types. The capsule gives the bacteria very good protection, so it protects them from drying out, protects them from the attachment of bacteriophages, which are viruses that specifically infect bacteria, and also protects them from phagocytic cells. So that's why human immune cells will find it difficult to deal with these bacteria. And the capsule also enables the bacteria to attach to surfaces.